Have you ever wondered about the silent warriors of modern medicine, the monoclonal antibodies, and how they are produced? These are not just tiny particles. They are our body's silent heroes, fighting off diseases and keeping us healthy. Monoclonal antibodies are a type of protein made in the lab that can bind to substances in the body, including cancer cells. They play a pivotal role in modern medicine, acting as the body's own defense mechanism. Their importance cannot be overstated. They are used in diagnostics to detect minute quantities of substances such as hormones, toxins, and cancer markers. Antibodies can also serve as therapeutic agents, directly targeting and eliminating disease-causing cells. Their precision and specificity make them a powerful tool in the medical world, giving hope to millions worldwide. Now you might be wondering, how are these powerful tools of medicine produced? Stay tuned to find out. The first step in the production of monoclonal antibodies is immunization. We begin this process with an animal, usually a mouse. This small creature is the starting point for our journey into the complex world of antibody production. To kickstart the process, the mouse is given an injection. Not just any injection, but one that contains the antigen of interest. Now, what is an antigen, you ask? Simply put, an antigen is a substance that triggers an immune response in the body, a response that includes the production of antibodies. Let's dive a bit deeper. Picture the antigen as a key. This key is unique and only fits into a specific lock. The body's immune system recognizes this foreign key and responds by producing a variety of locks, the antibodies. Each antibody is unique, designed to fit perfectly with the antigen key. This is the body's way of locking out and neutralizing the antigen. In the case of our mouse, the immune system is now busy creating a multitude of different antibodies to combat the injected antigen. It's like a tiny factory inside the mouse, churning out an army of antibodies, each with a unique ability to recognize and bind to the antigen. Now let's take a moment to appreciate this incredible process. The immunized mouse has geared up its immune system to react to the antigen, leading to the production of a variety of antibodies. But we're not interested in all of these antibodies, we're after a specific one. The challenge is to isolate the exact antibody we need from the multitude produced by the mouse's immune system. So the immunized mouse is now a tiny factory, producing a variety of antibodies. But how do we get the specific antibody we want? Well, this is where the magic of science takes the stage. Stay tuned as we delve into the fascinating process of cell fusion in the next scene. The second step in the production of monoclonal antibodies is cell fusion. This intriguing process is akin to a biological merger, where two cells become one. In the context of monoclonal antibody production, it involves fusing B cells with myeloma cells. Let's break it down. The B cells that we're focusing on are harvested from a mouse that has been immunized. These cells are essentially tiny factories, each one churning out a unique antibody in response to the immunization. However, while these cells are fantastic at producing antibodies, they don't live very long, which is a bit of a problem if we want to produce a lot of antibodies. So, we need a partner for these cells. And that's where myeloma cells come into play. Myeloma cells are a type of cancer cell that can proliferate indefinitely, but they don't produce any antibodies. It's like they have a superb production line, but nothing to manufacture. Now, we bring these two types of cells together in a process called cell fusion. When the B cells and myeloma cells are mixed together in the presence of a special agent called polyethylene glycol, they fuse to become hybridoma cells. These new hybridoma cells have the antibody-producing ability of the B cells and the longevity of the myeloma cells. It's the best of both worlds. But there's a catch. Not all cells will fuse, and some of the original myeloma cells will remain. To separate the wheat from the chaff, scientists use a special substance known as hat medium. Only the hybridoma cells can survive in this medium, effectively selecting them out from the mix. So, what's the result of all this? Now we have hybridoma cells, each producing a different antibody. But with all these different antibodies being produced, you might be wondering, how do we find the one we want? Stay tuned to find out in the next step of our journey into the fascinating world of monoclonal antibody production. The third step in the production of monoclonal antibodies is screening. This critical phase is like a treasure hunt, where the treasure is the hybridoma cell that produces the desired antibody. Since we've already fused our animal cells with myeloma cells, we've created a whole host of hybridomas. Now, it's a matter of finding the one that produces the antibody we're interested in. Sounds like searching for a needle in a haystack, doesn't it? But fortunately, scientists have developed some pretty ingenious methods to simplify this process. One such method is known as ELISA, which stands for Enzyme-Linked Immunosorbent Assay. This technique is like a litmus test for identifying our desired antibody. Hybridomas are cultured in separate wells, and the supernatant, the medium in which they are grown, is tested. The presence of the specific antibody can be detected by a color change, thanks to a secondary antibody tagged with an enzyme that triggers this change when it binds to our target antibody. Now imagine the ELISA as a grand stage where our desired antibody is the star performer. The stage, that's our well. The performer, our antibody, is invisible to the naked eye. But when the spotlight, our enzyme-tagged secondary antibody, shines on the performer, we get a visual cue, a color change, and voila, we found our star. But ELISA is not the only method. There are others like radioimmunoassay and fluoroimmunoassay. The choice of method depends on the specific requirements of the antibody in question. The key is to find an accurate and efficient method that will identify our desired antibody among the multitude of others. Through screening, we can now identify the hybridoma that produces our desired antibody. We found our star performer amidst the crowd. But how can we produce large quantities of this antibody? We'll explore this in our next step. Stay tuned to find out. The final step in the production of monoclonal antibodies is cloning and production. Now let's dive into this fascinating process. Once we have our hero, the chosen hybridoma that produces the antibody we're interested in, it's time for this cell to take center stage. The hybridoma cell is cloned using a process known as limiting dilution. This may sound complicated, but it's simply a method to ensure each new cell colony originates from a single cell. This way, we can be confident that all the cells in a colony are identical, or clones if you will, and they all produce the exact same antibody. Now picture this. You have a sea of identical cells, all working in harmony, each producing copies of the same antibody. It's like a perfectly choreographed dance on a cellular level. This, ladies and gentlemen, is the beauty of monoclonal production. But we're not done yet. We want these cells to produce the antibody in large quantities. So we move to the next phase, production. The cloned cells are transferred into a larger container, a bioreactor, and cultured under optimized conditions. It's like a five-star hotel for our cells, providing them with all the nutrients they need to grow and multiply, while also producing our desired antibody. Over time, the cells multiply, and the production of the antibody scales up. It's a bit like turning a small bakery into a large factory, churning out identical loaves of bread. Except here, our bread is the monoclonal antibody. Once the cells have done their job and produced enough antibodies, these are then harvested and purified. The end result? Large amounts of identical antibodies, all ready to be used in medical research, diagnostics, and treatment. And that, dear viewers, is how monoclonal antibodies are produced. It's a process that combines the wonders of biology with the precision of technology. The result is a powerful tool that's revolutionizing healthcare as we know it. So the next time you hear about monoclonal antibodies, remember the incredible journey these microscopic heroes have been on. Now that we know how monoclonal antibodies are produced, let's explore some of their applications. These antibodies have revolutionized the medical field, with roles in diagnostics, therapy, and research. For diagnostics, they're used in tests to detect specific antigens in blood samples, such as those for hepatitis and HIV. This precision allows for timely and accurate diagnosis. In therapy, monoclonal antibodies are designed to target specific cells, like cancer cells, without affecting healthy ones. This targeted approach has made treatments like chemotherapy more effective and less harmful. In the realm of research, these antibodies are invaluable. Scientists use them to study the intricate processes of cells and the immune system, leading to breakthroughs in our understanding of diseases and how to combat them. As you can see, monoclonal antibodies are incredibly powerful tools in modern medicine. They hold the key to understanding, diagnosing, and treating a wide range of diseases, giving hope to patients around the world. So there you have it, the fascinating process of monoclonal antibody production and its myriad applications. From the initial step of immunization to the complex process of cell fusion, screening, cloning, and production. These powerful tools are not only crucial in the field of medicine, but their use extends into diagnostics, therapeutics, and even research. They truly are a testament to the wonders of biotechnology. Remember, knowledge is power and curiosity is its fuel. Subscribe for more because you won't find me later. Stay curious, stay informed.